Hello again, everyone, and to those of you that are still joining us, and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. My name is Esther Obeng. I'm an assistant member in the Division of Molecular Oncology, and I will be co-hosting today with Dr. Marcia Wolarski, who's in the Division of Experimental Hematology. And we're very excited that Dr. Akiko Shimomura has joined us today. And so with that, it's my pleasure to introduce everybody to Dr. Akiko Shimomura, who's currently a professor of pediatrics at the Harvard Medical School and holds the Samuel E. Lux, the fourth chair of hematology oncology. Dr. Shimomura did her undergraduate training at Princeton University and obtained her MD and PhD degrees from the University of Rochester. She did her pediatrics residency training at Johns Hopkins and her pediatric hematology oncology fellowship at the Dana-Farber Cancer Institute and Boston Children's Hospital. I thought it was notable that she was actually the director of the bone marrow failure clinic at Boston Children's Hospital for several years before she was recruited out west, where she, in addition to advancing to the ranks of a full professor, was also the director of the bone marrow failure and MDS clinic at Seattle Children's Hospital. She returned to Boston back in 2015, where she directs the bone marrow failure and myelodysplastic syndrome program. As a trainee, when I was in Boston, interested in the genetics of bone marrow failure syndromes, Dr. Shimomura is one of the first translational investigators that I came across, both in reading her notes with very well cared for patients and also in clinical discussions about how to manage some of these complex patients. Dr. Shimomura's work has really led to the identification of molecular and genetic mechanisms causing bone marrow failure syndromes and MDS predisposition, and led efforts to develop appropriate strategies for surveillance, treatment, and prevention. Among her many formal accomplishments, Dr. Shimomura developed and directs the North American Schwachman-Diamond Syndrome Registry, which includes clinical data and clinically annotated biological samples. She also led a bone marrow failure and MDS clinical database and sample repository since 2001, and developed one of the earliest clinical genetic panels for bone marrow failure, and it, again, has identified several novel causative agents. Along with David Williams, she's the co-founder of the North American Pediatric Aplastic Anemia Consortium, or NAPAC, that has, again, developed and led studies impacting the treatment of pediatric patients with bone marrow failure syndromes. And with that, I don't want to stand in the way of Dr. Shimomura's talk entitled Clonal Evolution and Somatic Reversion in Genetic Predisposition to TP53 Mutated MDS. Thank you so much for joining us, Dr. Shimomura.
Thanks so much for inviting me to participate in this very exciting symposium. And it's a pleasure to be here, although I wish I could be there in person. St. Jude is where many of my very favorite hematologists and oncologists are, and it's so great to see everyone, even if virtually. So I'm going to share my screen. So today we'll be talking about genetic predisposition to MDS, particularly a form that predisposes to TP53 mutated MDS. And the way I got into this, as Esther so kindly described, is through bone marrow failure. And it's been long known that both acquired as well as genetic bone marrow failure conditions predispose to MDS. And indeed, more recently, the WHO has now recognized this new classification of myeloid neoplasms with germline predisposition. So although each individual germline predisposition condition is rare, taken as a whole, these comprise a significant subset of MDS, particularly in pediatrics, as shown so nicely by work of many of the investigators at St. Jude. I just wanted to, as an aside, since we have many oncologists in this call, and this is a cancer sort of symposium, that we are increasingly identifying patients with germline predisposition via somatic panel results. So many genes can be mutated either in the somatic or the germline context. And just because the panel is labeled somatic does not mean that the mutation arises somatically. It merely tells you that the mutation is present. And there are many reports, and here's a recent one, showing that in this case, about 30% of patients with RUNX1 mutated AML actually carried the RUNX1 mutation in the germline. And that, of course, carries completely distinct clinical implications for the patient, as we discussed actually earlier this morning. And I've just listed a few of these genes here. A couple of points to just keep in mind. In order to distinguish germline versus somatic, one can sequence non-hematopoietic tissues, such as skin fibroblasts, or if one tests family members and finds that the same mutation is running through other affected family members, it's tracking and co-segregating, that is very strong data to implicate a germline origin of the mutation. It's also important to recognize that variant allele frequency does not definitively distinguish germline versus somatic for a number of reasons, some of which I've listed here. So you have to be careful on trying to draw too many inferences from the variant allele frequency with respect to its implications. And then lastly, just a little caveat, which I know you have many experts in this topic at St. Jude, but I'll just mention that panels designed for somatic analysis should not be substituted and used to try to screen for germline mutations and vice versa. The panels are designed differently. They may tile different genes or different regions of genes that may or may not be appropriate for its opposite use. And the analytic pipeline is different for somatic versus germline. So do not use them interchangeably. However, if you do happen to detect a mutation on a somatic panel that can be germline, that should raise consideration of further workup. And additionally, if a mutation persists, even when a patient is in remission at the same variant allele frequency, that also warrants some consideration of whether that might, in point of fact, be germline. Now, one of the things that sparked my interest in doing a deeper dive into MDS was this paper from Coleman, Lindsley, and Ben Ebert, back when Coleman was in Ben's lab, where they were querying many patients who had samples in the CIBMTR because they had undergone transplant for MDS. And what Coleman found was that in patients less than 40 years of age, there was a significant subset of patients who actually had 2 SPDS mutations, and these patients had an abysmal prognosis. And this was associated with the presence of P53 mutations. So patients who had 2 SPDS mutations compared to those who had none or only 1 mutation. This is an autosomal recessive condition with Shwachman-Diamond syndrome. So you need 2 mutations to have the disease. Those with 2 mutations had 100% of them had P53 mutations somatically, which are, of course, a poor prognostic marker and may have contributed to their poor prognosis. So what is Shwachman-Diamond syndrome? It is well known to many of us who've trained in Boston because it was first named after Dr. Diamond, who was the chief of hematology at Boston Children's Hospital, and Dr. Shwachman, who was the chief of gastroenterology. And the patients classically present with cytopenias, particularly neutropenia, together with exocrine pancreatic dysfunction. So that's the digestive part of your pancreas. It secretes enzymes that are important for basically fat absorption. And so patients can present with failure to thrive, steatorrhea, but the presentation is highly variable, and their first presentation may be with MDS. So it's important to keep this in mind, that they don't always present in early childhood with cytopenias and pancreatic dysfunction. They can present later in life as teenagers or young adults with MDS, or as children with MDS. This is a multi-organ system disorder involving the skeleton, the liver, endocrine, immune, inflammatory, neurocognitive, but these may be clinically inapparent, and they can present with seemingly otherwise healthy people. The majority of cases are caused by biallelic or autosomal recessive mutations in the SPDS gene, which we'll talk about in detail in a moment. Additional genes have been described that also function in ribosomal pathways, which is what SPDS functions in. And these phenotypes are still being elucidated, but some of their phenotypic features can variably overlap with those of Shwachman-Diamond syndrome. The hematologic complications of Shwachman-Diamond syndrome are namely bone marrow failure and myeloid malignancy, AML and MDS. So we wanted to understand better because the outcomes were so abysmal with MDS. We thought, why don't we try to understand this better? And so in partnership with Kaz Myers, who co-directs the Shwachman-Diamond Syndrome Registry with me, she's based in Cincinnati, and our fabulous biostatistician, Edie Weller, and multiple colleagues listed here around the country, we pooled our data for 36 patients with Shwachman-Diamond syndrome who had developed MDS or AML. And we set out to characterize the clinical features and presenting features and outcomes. And I'm just going to cut to the chase, so there's a lot more detail in the paper, that outcomes with MDS were not great. Median survival was 7.7 years, whereas with AML, it was really very stark. The median survival was 1 year. There was only in this particular series one survivor. And the causes of death were from either treatment-related toxicities, TRM, or resistant disease. So what we concluded from this are our treatments don't work very well, and they're too toxic anyway. And so really what we need are better surveillance strategies to identify patients before they develop AML and intervene with the transplant prior to that time. We also looked at the question of marrow surveillance. How are we going to identify and risk stratify patients who might most benefit from transplant? And what we found was that only a small subset of patients were having any kind of marrow surveillance. The marrow surveillance was highly variable, with variable frequencies and variance in what specific tests were sent for a given marrow exam. And it varied not only by center, but it also varied from incident to incident, marrow to marrow for a given patient. And the median follow-up was similar in those who received surveillance and those who did not receive surveillance. And what we found, and this is just an observation, it's a description, we don't have the patient numbers to have robust statistics, but we observed that the median survival was 7.7 years in those with surveillance and 1.1 in those without surveillance marrows. So this gave us some pause as to what our surveillance ought to be, but gave us some hope that perhaps surveillance might be helpful, although we need more work. So, when we make a diagnosis of genetic predisposition to MDS before they develop MDS, it gives us an opportunity to intervene prior to the development of leukemia and prevent the leukemia. And we currently rely for our surveillance on blood counts. So we've shown, and there's an understandable preference for physicians to prefer blood counts over bone marrow exams. It's less invasive. But what we found in our Lancet hematology study was that blood counts were a very poor predictive marker for risk stratification. It did not predict very well patients who were at high risk of developing leukemia or MDS. By the time they developed their cytopenias, they had often already developed their myeloid malignancy. We also found that the severity of cytopenia did not predict risk of malignancy. Indeed, many patients who developed leukemia had stopped going to their hematologist because they only had a mild asymptomatic neutropenia and they thought their disease was mild and did not need to be followed until they developed their leukemia seemingly out of the blue, because they didn't have surveillance, so we don't know what led up to that. We also look at marrow morphology, we look at cytogenetics, FISH, and we also look at flow cytometry. And we can often detect signs of an impending leukemia. We can see high-risk clonal cytogenetic abnormalities such as monosomy 7 or the development of multiple cytogenetic abnormalities, complex changes. We can see new dysplasias arising in new lineages or progressive dysplasia in a given lineage. Or sometimes we'll see increasing marrow cellularity despite continued cytopenias. So you can start to get some hints, but and many patients were able in that way to be stratified and have a transplant prior to leukemia. But despite surveillance, there was also another subset of patients who, despite surveillance, went ahead and blasted off into leukemia a few months after their last surveillance marrow had shown nothing ominous. And we actually had those marrows evaluated in a blinded manner by our hematopathologists, our central hematopathologists who are very experienced. Tarek El-Ghattani, who worked with Blanche Alter for many, many years, and he's seen lots of these cases. And then Mark Fleming here at Boston Children's, who many of you know, was also highly experienced in these conditions. And there was no sign there. So what we know is that these are helpful, the marrow findings, but they're not enough. We need better surveillance strategies. So we turned our attention to somatic genomic analysis because this was at the time that we started this study coming into wider spread use. And we first collaborated with Dan Link at Washington University. And we had a cohort of patients with severe congenital neutropenia, cyclic neutropenia, and Shwachman-Diamond syndrome. And we just took sort of a snapshot in time at that time of samples of patients who did not have malignancy. And what we found was in the patients with Shwachman-Diamond syndrome, a significant proportion of patients actually carried pathogenic P53 mutations. This is in marked contrast to patients with severe congenital neutropenia, which also carries a high risk of myeloid leukemia, but these patients did not carry somatic mutations in P53. These patients actually develop somatic mutations in the CSF3R gene. Cyclic neutropenia patients do not have an elevated leukemia risk, although that is currently under further study, and then healthy controls. It's very rare in healthy children to have clonal hematopoiesis. So when this came out, we received a lot of questions. What should we be doing with these patients with P53 mutations? And our answer was, we don't know. In order to understand the clinical implications of such mutations, we needed a longitudinal cohort study with clinical outcomes. And so we turned our attention to the Shwachman-Diamond syndrome registry, which I had founded many, many years ago and was subsequently joined by Kaz Myers at Cincinnati, and she's been a fantastic partner, a hugely talented investigator and transplant expert. And we systematically collected samples and clinical records. And I started this whole endeavor around the year 2000. This was before we even had the technologies to ask the kinds of questions that we could now, but we knew that this was coming. So in the registry, it was designed to collect both marrow, blood, and fibroblast samples so that we had paired sort of marrow and then non-hematopoietic tissue collected longitudinally over many years. So then we used the registry then to ask the following questions. What are the mechanisms driving clonal hematopoiesis in bone marrow failure and MDS predisposition? What are the biological and clinical consequences of recurrent somatic mutations? And can an understanding of this biology inform a conceptual framework to inform the development of rational surveillance strategies? So this work was done in close collaboration with Coleman Lindsley, who is an expert, of course, in somatic genomic analysis of myeloid malignancies, and Alyssa Kennedy, who was a fellow and is now an instructor in my lab, who also actually worked for a while early in her career at St. Jude. We were joined in this endeavor by Chris Gibson and Nick Camarda in Coleman's lab, as well as Scott Carter at the Broad with his trainee Robert Klein, who Scott was an amazing computational biologist who developed very nice algorithms to do these kinds of analyses, including copy number variants. And we started with a discovery cohort where we did deep whole exome sequencing of paired marrow and fibroblast samples. We queried 29 patients with Shwachman-Diamond syndrome caused by biallelic SPDS mutations with a variety of clinical types. So some patients had MDS, some had AML, 2 had severe marrow failure defined as needing to go to transplant, and 15 did not have severe marrow failure, MDS, or AML. They were sort of at baseline. We identified recurrently mutated genes and then used this information to inform the design of a targeted custom panel that also included recurrently mutated genes that were found in myeloid malignancies. We then went on to sequence paired marrow and fibroblast samples for each patient for 110 SDS patients, most of whom had SPDS mutations, for a total of 302 samples with defined mutation, germline mutations. We also took 11 patients who were diagnosed as SDS-like. There is this entity that has been defined by a consensus conference many, many years ago of patients who share clinical features, have clinical features of Shwachman-Diamond syndrome, but we cannot genetically define them. So these were patients who lacked any of the known SDS genes mutations, and we also queried samples from these patients. And so what we found, let me take you through this. Each column is an individual patient. Each row is a different gene. At the top, we have survival or death is in red. And we have here their clinical phenotype. So these are the SDS patients here. These are the SDS-like patients here on the right. These are patients with no severe marrow failure. These are the patients with severe marrow failure. These are the patients with AML and MDS. And you can see right off the bat a lot of blue. These are mutations in a gene called EIF6, which were found in patients with and without bone marrow failure. We also found P53. They were found in all patients with AML, some with MDS, and many with bone marrow failure. Without severe bone marrow failure, but did not have a myeloid malignancy. There was a statistically significant association of P53 with malignancy, whereas the EIF6 frequencies were not statistically correlated with malignancy or without malignancy. The patients with severe marrow failure, interestingly, did not have identified somatic mutations. And interestingly, these SDS-like patients also lacked clonal hematopoiesis, suggesting that these patients are biologically distinct. We also looked at cytogenetic clonal abnormalities. These are those without myeloid malignancy. MN is myeloid malignancy, and these are those with. And you can see that del20q or monosomy 7 is found in patients with MDS or AML, as are some of these other chromosomal abnormalities. Del20q is quite common with or without myeloid malignancy, and it's a common mutation in Shwachman-Diamond syndrome, and we will get back to that in a moment. However, what I will say is the del20q clone was not the malignant clone. Even if del20q was found in patients with leukemia, the del20q clone was separate from the leukemic clone. Not only were EIF6 and P53 mutations commonly found in these patients, but within a given patient, there was a propensity to develop multiple EIF6 or P53 mutations. So this is mutations per patient. This is a patient with more than 10 EIF6 mutations. This is a patient with 15 P53 mutations. The other two recurrent mutations were PRPF8 and casein kinase. And as you can see, there were some patients who also acquired recurrent mutations there. But I'm going to focus the rest of my talk on the most common mutations, but don't forget about these. Now, it turns out that the pattern of clonal hematopoiesis in Shwachman-Diamond syndrome was really quite distinct from that which we see in aplastic anemia, right, or CHIP or MDS. So here in Shwachman-Diamond syndrome, 97% of the mutations were found in these four genes that I just mentioned, particularly EIF6 and P53. In contrast with aplastic anemia, we see more PIG-A, B-Core, B-Core 1, DNMT3A, ASXL1. With CHIP, of course, DNMT3A is huge, and you can see some of these others, TET2, ASXL1. And then once you get into adult MDS anyway, splice factors, TET2, etc., are more common. So it was really a quite distinct pattern that appears to be driven from this particular germline mutation. There was nothing particularly striking about the types of mutations we saw. The C to T transition mutation was quite common in both EIF6 and P53. This is, of course, the common deamination transition that we see often with age-associated clonal hematopoiesis as well. And there is a striking correlation between clonal hematopoiesis and age, but now at a very young age. So normally, of course, we do not see clonal hematopoiesis in children and young adults to any particularly high extent. But now you can see that there's a correlation between age and number of somatic mutations. And if you look at the proportion of patients with clonal hematopoiesis at different ages, those who were between the ages of 0 to 10 had over half of them already had developed clonal hematopoiesis, and clonal hematopoiesis was universal in all 10 of our patients who were greater than 21 years of age. It's interesting to look at, just descriptively, the association between age and hematologic complications. So severe bone marrow failure was observed in our cohort at an early age. And this was also observed not only in the Schwachman registry, but also in the French neutropenia registry, also noted that severe marrow failure tends to occur at younger ages in patients with Schwachman-Diamond syndrome. In contrast, MDS and AML tends to occur a little bit later, still young, but in the sort of people past the age of 10 into young adulthood. And similarly, clonal hematopoiesis was more common in the older age groups compared to those patients who lacked clonal hematopoiesis. And this was interesting because we looked actually at hematologic parameters as a function of age. And this was done in collaboration with many people, including friends at St. Jude. And you can see that there's a striking drop very quickly in bone marrow cellularity that then kind of levels off. But in contrast, the blood counts actually tended to, in this case, the neutrophil counts, there tended to be an improvement in the neutrophil counts as a function of age over time, which is a little bit different from the way we typically think of bone marrow failure. You kind of think of, as your cellularity becomes very low, you are more inclined to have a progressive drop in your blood counts, but that's not what we saw. And it was interesting to note that this increase in blood counts correlates, at least age-wise, with acquisition of clonal hematopoiesis. Whether that's linked or not requires additional work, but it was an interesting observation. So what are the functional consequences of recurrent somatic mutations? Well, we turned then to understand the implications of EIF6 and P53 mutations in patients with Schwachman-Diamond syndrome who have a predisposition to P53 mutated MDS and AML. We turn to work from Alan Warren's group, and Alan has done really nice work delineating the role of SPDS in ribosome biology. So SPDS is bound to the pre-60S ribosomal subunit, and it is required for ejection of EIF6 from the pre-60S subunit. And EIF6 binds to the 60S subunit at the interface between the 60S and the 40S subunit and sterically inhibits the joining of the 60S to the 40S subunit. That was described by other people. What Alan found was that SPDS acts to allosterically cause conformational changes that result in eviction of EIF6 via the EFL1 GTPase. So what you can see here is that if you are lacking SPDS, if you're deficient in SPDS, as are patients with Schwachman-Diamond syndrome, you can no longer eject EIF6. So now you've got EIF6 kind of stuck to your 60S ribosomal subunit, and this subunit cannot form a mature, translationally active ribosome. So we first set out to map these mutations in EIF6, and as you can see, they're located throughout the EIF6 protein structure, and they're found across different domains of the protein. And the types of mutations are annotated in different colors. Red are missense and in-frame deletions, and the gray balls are truncating mutations. And as you can see, there were different types of mutations up here. Missense was very common, in-frame deletions less common. Nonsense mutations, frame shift indel, splice site, and stop loss. So you can see that there are certainly clear mutations that would be predicted to cause loss of function. And the most common mutations, the size of the ball is proportional to the number of mutations. The most common mutation was an N106S variant, followed by the R96W variant. So we set out to then ask what were the functional effects of these mutations on the EIF6 protein structure, stability, and function. And so first we had to model EIF6. There were no great models of human EIF6. So this was modeled by Chris Ball over at the Protein Design Institute for Protein Innovation at the time. And he was joined by his postdoc, James Bowman. And you can see here in color, sort of mapped onto this structure, are the mutations, and the color denotes the predicted delta delta G changes that would be predicted based on these mutations. And these thermodynamic changes are graphed here. You can see that most of them are predicted to destabilize the protein. However, there is a subset of variants that were located at the site of the interface between EIF6 and the 60S ribosome, and these were not predicted to destabilize the protein, but might perhaps affect the interaction between EIF6 and the 60S ribosome. So Alyssa set out to functionally query this in my lab, and she expressed wild-type EIF6 as well as a series of mutants, and showed that although the transcript levels for all of these were similar, the protein products were markedly lower in many of these mutations. And indeed, these were the mutations that were predicted to be destabilizing based on the energy calculations on the previous slide. And she found through data that I'm not going to show you that if she added a proteasome inhibitor, she could restore protein expression. So this appears to be a destabilization of the protein. The most common mutation here, the N106S mutation, as you can see, was not destabilized and expressed levels similar to that seen with wild type. So we have two classes of mutations, those that are destabilizing and those that aren't. So we queried the N106S mutation. So this asparagine at residue 106 is predicted to be important in the interaction between EIF6 and the 60S ribosome. So we asked the question, does this mutation affect the association of EIF6 with the 60S ribosome? And to do that, we made protein lysates and ran them through a sucrose gradient. This is the top of the gradient, this is the bottom of the gradient. And we blotted for either EIF6, which will include endogenous EIF6, and as you can see, it migrates down, it co-sediments with the 60S subunit, as you would predict. But we also expressed a exogenous wild type or N106S mutant EIF6 that carries a V5 tag. So we could actually blot for the V5 tag and showed that the wild type V5 tagged EIF6 co-sediments with 60S. But in contrast, the N106S variant now is here in the free fraction at the top of the gradient with the free proteins and is not co-sediment, is no longer associated with the 60S ribosomal subunit. So this abrogates the association of EIF6 with 60S. We also found that this mutation results in mislocalization of EIF6. Normally wild-type EIF6, and we both looked at endogenous EIF6 as well as tagged, V5 tagged EIF6, localizes in the nucleus, and particularly in the nucleolus. And in contrast, so it's here in green. And here you can see that when you introduce the N106S variant, it is now no longer localized to the nucleolus, which is marked by this red fibrillin marker. And we quantitated this in a blinded fashion and showed that N106S is no longer localizing to the nucleolus. So this mutation impairs its intracellular localization. We then wanted to ask, what were the effects of mutations on ribosome biology? So we looked at the relative assembly of ADS subunits in control versus SDS patients. This is old work published back when I was in Seattle in Blood. And we and others, including Alan Warren's group and others, have shown that in patients with Shwachman-Diamond syndrome, there is a decrease in the amount of ADS subunit produced, this is a sucrose gradient, compared to the 60S subunit. And you can actually calculate the ratio, in this case, the ADS-40S ratio, but you can also do ADS-60S ratio. And you can see that compared to controls, there's a decrease in the ADS subunit compared to the 40S or 60S. So we then looked at the effect of knocking down either EIF6 or P53, right? These are the most commonly mutated genes in in patients with Shwachman-Diamond syndrome. And as you can see, with knockdown of EIF6, you can see an increase in the amount of ADS subunit that can now be assembled in cells from Shwachman-Diamond syndrome patients. And I'm showing you the actual gradients here. It's just a representative gradient. So if you compare the height of the 6DS versus the ADS, you can see that there's much more ADS relative to 6DS following knockdown of EIF6. In contrast, when you knock down P53, there's no significant increase in the ADS subunit. So it doesn't rescue, P53 does not rescue the ribosomal abnormality in Shwachman-Diamond syndrome. We also knocked down EIF6 and or P53 and looked at protein translation. And this is using an assay called OP-Puro. So this is a puromycin analog that, like puromycin, can bind to and basically halt the protein translation of nascent polypeptides. So it covalently sort of blocks peptidyl transferase, and through click chemistry can be covalently bound to a fluorophore that you can then use to quantitate the amount of peptide, active peptide synthesis that was ongoing. And so you can see here, if you look at cells from patients with Shwachman-Diamond syndrome, in black is our control, where we just used an SH construct against luciferase, versus an shRNA knockdown targeting EIF6. And you can see that there's a clear shift. This is a log scale. There's a clear shift to the right of OP-Puro intensity, consistent with increase in protein translation. So it's rescuing the protein translational defect that we see in Shwachman-Diamond syndrome. In contrast, knocking down P53 really had no significant effect, if any, on protein translation compared to luciferase control. Both EIF6 and P53 mutations alleviated checkpoint activation, here mediated by CDKN1A expression. So both of them could, so when you knock down SBDS, or if you look in SDS patient cells, there is an activation of checkpoint pathways, and this can be alleviated by knockdown of either EIF6 or P53. Lastly, we looked at the effect of P53 on hematopoiesis. So, this is Shwachman-Diamond syndrome is a bone marrow failure condition. So if you knock down SBDS, you get decreased hematopoiesis. And if you add back wild-type EIF6 is deleterious, right? It's going to inhibit ribosome formation. So that knocked down hematopoietic colony formation of both myeloid and erythroid. However, if you add in, with lentivirus's mutant EIF6, there was no significant effect on colony formation in the Shwachman-Diamond syndrome model. And similarly, we did it with N106S. In contrast, P53, if you knock that down, will rescue the colony-forming defect in Shwachman-Diamond syndrome. So this was intriguing because Alan Warren had previously published using this yeast model of Shwachman-Diamond syndrome, sort of a genetic rescue, a spontaneous genetic rescue that he observed following sort of outgrowth of these Shwachman yeast model, which normally grow very poorly. But if they acquire mutations in TIF6, which is the yeast equivalent of EIF6, you could see that growth was restored. And if you look, you can even see here the N106 residue here, and many of these residues were mutated in patients as well, lending further credence to this idea of somatic genetic rescue of the Shwachman deficient cells. So although Shwachman-Diamond syndrome is rare, I think this illustrated some general principles that are more broadly applicable. So here you've got a germline genetic stressor, in this case, a SBDS mutation. This leads to stresses that are specific to the mutation in question, in this case, a ribosomal abnormality, causing ribosomal stress due to impairment of ribosome formation because of EIF6 retention. This, in turn, ribosomal stress signals to checkpoint activation and reduces cellular fitness of hematopoietic cells. In contrast, either EIF6 or P53 mutations can rescue the hematopoietic fitness defect, right? It increases fitness of hematopoietic cells and it decreases checkpoint activation, but it does so through completely different mechanisms with distinct clinical effects. Here, the EIF6 mutation actually rescues the underlying ribosomal stress affected by the germline SPDS mutation, and this is actually a somatic adaptive mechanism for rescue of the ribosomal abnormality. In contrast, P53 mutations do not improve ribosomal stress, that's still there, but it merely abrogates the anti-oncogenic checkpoints to improve cellular fitness, but the stresses continue. Right? So it's a maladaptive somatic mutation. In thinking about this, we also turned our attention to the well-known del20q clonal abnormality that's frequently observed in patients with SDS, and we published in Blood Advances. This is actually the most common mutation, and other groups have observed this as well. Yigal Dror in Canada was among the earliest to observe this with Mel Friedman, the French neutropenia group, the Italian neutropenia group, the UK group. So I want to give a lot of credit here, but the Italian group really did a lot of work on this. And what they found was that the deletion, and they mapped specifically the region of the deletion, and they found that that EIF6 was deleted in all cases of del20q that were observed in Shwachman-Diamond syndrome. So that suggested but did not prove that EIF6 was important. I mean, this is a multigenic deletion, right? But now with the observation of specific mutations within EIF6, that really narrowed the focus now to EIF6 in this del20q clone. And shortly after our paper came out in Nature Communications, an independent group also published similar observations. They found EIF6 mutations were recurrently mutated in an independent cohort of patients with Shwachman-Diamond syndrome. This is Patrick Reby's work with Alan Warren. And they found that if they introduced EIF6 mutations into these Drosophila. So this is a Drosophila model of SPDS. So it's a very severe phenotype. This is a wild type larva at 1, 3, and 5 days. And by 11 days, you have this nice fly. In the Shwachman model, where they've introduced 2 mutations, it gets stuck. It's lethal at the larval stage, and the larvae don't develop nicely. And in contrast, when you introduce now this specific mutation, you can now restore formation of a fly. And they introduced lots of different EIF6 mutations. Some of them were more effective than others at rescuing. The degree of rescue varied, but this was proof of concept that this can rescue the phenotype. So we then wanted to ask, we see P53 mutations, we see EIF6 mutations. Do these cooperate to drive clonal progression? Why do you need both? And so this is just the results of bulk sequencing of several individual patients. And in blue are the EIF6 clones. This is the variant allele frequency. And in red are the P53 clones, and then in black are others. And you can see that they had most patients had both EIF6 and P53 mutations. So the question was, do they cooperate? So to ask that question, we did single cell sequencing and found that each panel is an individual patient, each clone is an individual row, and each mutation is an individual column. And the P53 mutations are in red, the EIF6 mutations are in blue. And as you can see, you never see red and blue mutations in the same clone, indicating that the EIF6 mutations and the P53 mutations arise in independent parallel clones. Not only that, each multiple EIF6 mutation arises independently. So you've got lots of different independent EIF6 clones and P53 clones arising in these patients. And I'm just showing you a few representative samples here. We never saw an EIF6 clone that subsequently developed a P53 mutation. So those appeared to be mutually exclusive at the single cell level. We did, however, see a patient who had originally had a parental P53 clone that subsequently acquired an EIF6 mutation. So we did see P53 clones develop EIF6 mutations. And we did see in one case a patient who had an EIF6 mutation who subsequently developed a TET2 mutation. So, it's complicated. But by and large, I would say that we did not observe, at least in our cohort, co-occurrence of EIF6 mutation that subsequently developed a P53 mutation. So we also looked at the clonal dynamics. What happens to these clones once they develop? And so you can see here that the variant allele fraction for most of these mutations were low, right? Less than 0.1. But there were some that were higher. This is a log scale, right? So some are higher. We also looked at their variation over time, and by and large, there was not a lot of variation over time for either EIF6 or P53, although there were some exceptions with EIF6. And indeed, I've seen with del20q, I have some patients who have developed 80% of their cytogenetics carry the del20q clone. And the del20q clone tends to persist. That was published in the Blood Advances paper that I just referenced. So we tried to look in more granularity at clonal dynamics to understand, what do these mutations mean for patients? What is the prognostic significance of them? So these are young patients, and I'm just showing you a representative 4 of them here. Each arrow represents a time point at which we sampled their marrow. Each clone is represented by a different line, and blue represents EIF6 clones and red are P53 clones. And as you can see, early in life, we typically did not see, they were not born with these mutations. You can find earlier marrow samples without these mutations. They're typically acquired after birth. And you can see that, you can start to see these clones kind of developing, and only a few of them develop early on, right? But if you look later, so these are the older patients, in their 20s, 30s, late teens, you can see that by now they've developed multiple clones, EIF6 clones and P53 clones. But by and large, these remain stable, right? Over many, many years. In some cases, we're talking, very close to a decade of follow-up with no change in the clone size. They're just kind of sitting there at low variant allele fraction. So the presence of a P53 mutation per se did not portend, rapid outgrowth or leukemia in these patients. So what then distinguishes clones with a high risk of transformation versus those likely to remain clinically stable? Because most of these are clinically stable. So to ask that question, we first turned our attention to those patients who developed myeloid malignancy. And all 7 patients who had AML had biallelic alterations of P53, either through acquisition of a mutation of the second allele, a P53 mutation plus a deletion, or copy-neutral LOH. There were 4 patients with MDS who lacked P53 mutations, so this may not be the whole story. So there's more to look at here, but the biallelic P53 mutations were found in all patients with AML leukemia. So the question was, is it too late? By the time you develop a biallelic P53 mutation, have you already developed your leukemia, and is it too late to intervene? And so to start to answer this question, and it's still an open question, but I'm going to share this one informative patient with you who gave us some hope, is a patient who had many, many years of surveillance. And this was the results of her bulk sequencing. And you can see that the clones are followed with lines. The red are the P53 mutations. The blue are the EIF6 mutations. And the black is the casein kinase mutation. And you can see just a few months before she blasted off into AML, she had a marrow done that showed no sign of anything impending. Her blood counts were stable. Her marrow looked fine. I actually had her marrow reviewed by our hematopathologists, and they did not see anything ominous in that marrow. And the clone that would eventually become the leukemic clone, this is a log scale, was lurking there, but at a variant allele fraction that would not even be caught on many of the commercial somatic clinical panels, right? So we then did a deep dive and looked by single cell analysis at multiple years of surveillance. And this is just year 6 of surveillance, just before she blasted off into leukemia. And as you can see here, she again had multiple clones of EIF6 and P53. But I draw your attention to this clone. And if you look, I'm going to sort of blow this up here so you can actually see it. She had most of her P53 mutations were heterozygous, right? And I forgot to point out on all the single cell analyses that we see in those clones that persist over time, they are heterozygous mutations in P53 or EIF6. But you can see in this patient, over 6 years before she developed her leukemia, she didn't have this clone. This is the eventual leukemic clone. But, about 4.5 years before she developed leukemia, you can see this little tiny homozygous P53 clone lurking here that then had risen to 3.9% variant allele frequency just before, several months before she blasted off into AML. So this gives us some hope that perhaps this strategy to try to incorporate P53 analysis into surveillance may be helpful for risk stratification and identification of patients who are most likely to benefit from early transplant. So let's get back to our original title of this talk, which was Somatic Rescue versus Clonal Evolution. And so I think the concepts here illustrated by Shwachman-Diamond syndrome are broad principles that can be generally applicable. So upon acquisition of a germline mutation, you have a reduction in cellular fitness. Now you have a marrow with hematopoietic stem cells functioning in a failing environment, right? The other cells are also failing. You've got ribosomal stress and checkpoint activation. And there is huge selection pressure and outgrowth of the acquisition of any mutations that confer a fitness advantage. And in this case, the fitness advantage can be obtained through somatic compensation to actually rescue the underlying germline abnormality, in this case in ribosome function, but the checkpoints, the anti-oncogenic checkpoints remain intact. And you can do that either through point mutations or deletion of 20q to effect functional EIF6 haploinsufficiency. In contrast, P53 inactivation, initially through heterozygous and subsequently biallelic P53 mutations, unfortunately improve fitness and improve hematopoiesis, but drive towards malignancy. And so we can then, the implications here are perhaps EIF6 is a attractive therapeutic target to potentially both improve hematopoiesis, which we want to do, but simultaneously reduce leukemia risk, right? We saw none of the cells that had EIF6 mutations went on to develop leukemia, and none of them acquired P53 mutations. So perhaps it's protective in a cell-intrinsic manner. In contrast here, perhaps we can utilize this observation of P53 inactivation and clonal evolution to help inform risk stratification and identify those patients most likely to benefit from early intervention with transplant. So I'd like to just I tried to thank everybody as we went along, so I'm not going to take a lot of time here except to thank my lab, who's fantastic, my clinical research team led by Maggie Malsh, my partners at Cincinnati for the registry. I thanked everyone. Christian gave us the microRNA constructs for modeling SDS, and Stefan gave us the P21 reporter cell line that we used for that assay, thanking our funding agencies and all of our collaborating physicians, patients, and families. So thank you very much.
Thank you, Akiko, very much for this outstanding lecture, which perfectly shows how years, or I would say rather decades of hard work and a lot of stamina on registry building eventually leads to such important molecular discoveries. And this, of course, in the long term leads to a more informed patient surveillance and improved outcomes. So it's almost top of the hour. So in 3 minutes at 1 p.m. Central, we will close the official event, but we would like to invite all of you who are interested to stay on for a continued discussion in the extended Q&A session, which will take half an hour until 1:30. And before we get back to the questions, I would like to thank you all for coming one more time and encourage you to visit our website and mark your calendar for the upcoming talks. I have posted the link in the chat box right now. So next week, February 24th, Dr. Ralph de Bardinis of the Children's Medical Center Research Institute at UT Southwestern will be presenting on human metabolic outliers. And now I would like to come back to our questions. So the first question asked by Dr. Sushi Sahu, who is also interested in clonal hematopoiesis. She's a postdoc at St. Jude and interested in clonal hematopoiesis in pediatric MDS. So she asks if therapeutic targeting of IF6 might be potentially considered in Shwachman-Diamond syndrome to rescue the phenotype.
So thank you for that question, Sushi, and great to see you. You're also doing fantastic work with Marcin. So the short answer is yes. So we are, of course, pursuing that through a variety of ideas. I know that there are other groups that are also looking at that. Stefano Bifo, who is one of the world experts in EIF6, published a paper of a chemical library screen, and he found some potentially interesting compounds, although it was a very preliminary paper and there weren't a lot of details in it, but that was exciting. Alan Warren, I know, has something in hand that he's mentioned, but no details are available, so we're also excited about that. So I think that this would be novel, and perhaps a shift in our paradigm for these bone marrow failure and leukemia predisposition syndromes, if we can find agents that both improve the bone marrow failure and prevent leukemia. Right now, we rely on things like GCSF, which can improve, for example, the neutropenia, but does not reduce and perhaps might increase, though we're not sure about that, leukemia risk. So this would be a nice approach. We're getting pretty good. Thanks, I can't claim credit. It's our transplant colleagues who have really improved transplant to the point where bone marrow failure outcomes are excellent. Transplant outcomes for bone marrow failure are superb. All transplants have potential risks, but as you can see, the transplant outcomes for leukemia are horrendous. So we clearly need to prevent the leukemia or come up with better ways to treat it. Hopefully both.
Thank you. So another question comes from Dr. Jinkui Zhang, from director of our computational biology department. The question is about if for the clonal hematopoiesis mutations detected in Shwachman-Diamond patients, have you also profiled normal individuals? So I think the marrow specimens with matching age to see if these variants can also be present.
Right. So there was an early paper from Welch and Tim Lay, and they looked across ages at clonal hematopoiesis using different types of techniques. And clonal hematopoiesis is very rare in healthy children. So we did not query that here. What we're finding, though, is that in patients with inherited bone marrow failure conditions, clonal hematopoiesis is quite common. And then even with acquired aplastic anemia, so this is work from Neil Young and Seishi Ogawa and Jarek Masajewski, combined their cohorts in a lovely New England Journal paper. It was primarily adults, though, but they had some younger people there. And with you can get clonal hematopoiesis with acquired aplastic anemia as well. So it may be a combination of some of the driver appears to be dependent on the germline mutation, because as I mentioned, depending on the germline mutation, you get different types of somatic mutations growing out. With Elaine mutations, you get the CSF3R mutations. With GATA2, you see different mutations with ASXL1 and some of these other things. So the driver mutations, the underlying germline driver mutations do, I think, direct to some extent what types of clones have a selective advantage and tend to grow out. But in children, it's quite rare to see clonal hematopoiesis. And there's some unpublished data that I can't share, but that would corroborate that. It's not my data, that's why I can't share it.
Thank you. So along the same lines about the driver mutations, another question from Dr. Masanori Yoshida is about the additional mutations you discovered in the casein 1A1, the CSNK1A1 and PRPF8, do you think that these events might be involved in adaptive or maladaptive clonal hematopoiesis in Shwachman-Diamond syndrome?
It's a fantastic question. We need to study those genes. We have not really queried in any detail those genes, though, in collaboration with Coleman, we are doing a deeper dive into the functional consequences of those mutations. So I think that will be important. So I don't think it's all just going to be about P53, and you're making a good point that we need to keep an open mind and look more broadly.
Thank you. We have more questions coming in from across the ocean. So Henry Kassler is hailing from Denmark, asking a question about if the clonal development in Shwachman-Diamond syndrome could be monitored in peripheral blood specimens, or do you need to do regular bone marrow sampling?
That's a fantastic question. Thank you, Henrik. It's good to see you. So we don't have data to address that, though we're trying to put a project together now. But the best data that addresses that is actually published in Blood Advances from Annette Kim, and I almost included that in the talk, but I ran out of time, where they compared like 167 or something like that paired samples, blood and marrow, from patients with hematologic malignancies, and found that patients who at diagnosis had paired samples had excellent concordance between marrow and blood. And one might imagine that at diagnosis, you have a lot of circulating leukemia burden, perhaps. But for patients in remission, where you're trying to look at MRD, sort of like what we're trying to look at for patients who haven't developed malignancy yet, but we're trying to look at some early sign that something might be lurking there. There was less concordance. And even in some cases, and she published some detailed analyses, there were some patients with actually a pretty high VAF P53 variant found in the marrow. I think it was like 30% VAF or something like that, that was missing in the blood. And so I just want to be careful. I've not made the recommendation yet to my patients to do this only in blood, partly for that reason, because I'm not sure how sensitive the blood is to predict impending malignancy. One might imagine that if a mutation were acquired in an early progenitor or stem cell that doesn't allow it to differentiate well and get into the periphery, that might still cause problems in your marrow, but not be detectable in blood. And secondly, in marrow, we can also see additional signs such as increasing dysplasia, increasing cellularity, that despite, persistent cytopenias, clonal cytogenetic abnormalities, which we don't detect so well with the somatic mutation analysis. So I have a lot of hesitation before recommending that, but of course we don't want to keep doing regular bone marrow. So we hope we can move there someday.
Thank you, Kiko. Another question from Lawrence Baker is about if you have examined patients with Li-Fraumeni syndrome who have loss of function germline P53 mutations who developed AML.
So that's a fantastic question. So that's been done by others, and it needs more work, but one of the big take-homes from prior studies was there is a tendency to acquire a mutation in the second P53 allele in the different types of mutations that arise in Li-Fraumeni patients. So it would sort of go along with the paradigm that we're seeing in Shwachman-Diamond syndrome, except they're not born with the P53 mutations, they acquire them. And certainly work from our adult colleagues such as Eli Papa-Emmanuel and Ross Levine and others are showing that biallelic P53 mutations, even in adult MDS AML, carries a worse prognosis than the heterozygous P53 mutations.
Thank you. One question that we have is about the surveillance. So from Jason Farrar from University of Arkansas, he is congratulating on the great talk, and he wonders if you got a sense of kinetics of the clonal changes from your patient cohort. Do you have a sense yet of optimal timing intervals for these surveillance marrows?
That is a fantastic question, and we were hoping that the somatic genomics would give us a sense of that. So it's early days yet for me to make a recommendation, but Right now, for lack of better data, what we're recommending is what we recommend for patients with Fanconi anemia, which is another high-risk sort of leukemia condition, predisposition condition, and we recommend annual marrows. But as you might imagine, there are a lot of families that don't want to do annual marrows. There are others who want to do much more frequent marrows, like some parents are really nervous about the leukemia and they would want me to marrow all the time, so we kind of have to pull them back. We do blood counts in between, but there are some families that say, well, what are really the data? And we just acknowledge we don't have data. And we partner with the family to try to come up with an acceptable frequency of these marrows. And I do think that a baseline marrow is very helpful because it gives you a sense of how worried you have to be going forward. So you've got a baseline and you also have something to compare it to. If the marrow looks completely fine and there's nothing worrisome there, and the blood counts are stable, sometimes and they're stable in a good range, sometimes I'll let them go, more than a year, 2 years, 3 years, but they have to acknowledge I won't be able to monitor there. We have had some patients who blasted off, right? But now we hope that with the addition of somatic genomics, that will help. I have some patients with their marrows are clean. I can't find anything because they're young in their marrows by somatic genomics, by, their dysplasias are stable and the cytogenetics are clean, their blood counts are stable. Those are the ones I worry a little bit less about and can be a little bit less aggressive. So we'll just have to see. We don't have great answers though.
Thank you. We have another question by Dr. Jun Yang from our Pharmaceutical Sciences Department. It's on the origin of leukemia in Shwachman-Diamond syndrome. So if the effects of germinal defects are compensated by somatic mutations, such as IF6 or TP53, how do the germinal mutations, the German Shwachman-Diamond gene mutations, contribute to leukemia pathogenesis? Or maybe the main consequence of germinal and Shwachman-Diamond mutations is to actually prompt acquisition of somatic P53 alterations, which will then lead to leukemia.
It's a fantastic question. So we think EIF6 is compensatory. So it rescues the SBDS phenotype and does not actually contribute to leukemia and may, in point of fact, be protective against leukemia. But the P53 mutation, it's a great question. And we're trying to query some of the functional effects of the ribosomal abnormality in SBDS deficiency because we don't actually understand what specifically about this is deleterious. Is it just that it's ribosomal stress? The reason I don't think so is Diamond-Blackfan anemia also exerts ribosomal stress, right? It exerts ribosomal stress in a similar way. It's not a joining defect, but you're actually missing a ribosomal component. You're haploinsufficient. And that can also trigger P53 activation. And P53 is hyperactivated in patients with Diamond-Blackfan anemia, yet they do not have a high propensity to develop leukemia the way the Shwachman patients do. As a matter of fact, it's so rare, we don't even do regular surveillance in those patients. And it's unclear whether their malignancies are P53 driven, at least their blood malignancies. They do develop osteosarcoma, which is interesting because that's a typically P53 driven malignancy, but the studies are still not out yet. So I don't know if it's just ribosomal stress per se, because it's so different with DBA, which is also a similar type of ribosomal stress. So it may be combinatorial, but time will tell. Great question.
Thank you. I have a similar question along the same lines on leukemia development. So some of the Shwachman-Diamond patients have severe neutropenia and require GCSF therapy. So do you think that GCSF might reduce the stress on the marrow or might actually increase the propensity for developing such mutations, the bad mutations?
So it's a great question. In the registry, we've not been able to, so we actually had data, which is buried in the supplement, that a subset of patients who developed AML had received GCSF, but GCSF therapy is actually quite rare in Shwachman-Diamond syndrome. And in the graph that I just showed you, the neutropenia tends to be most severe in the young children, interestingly, the babies, and then it often gets better. And indeed, the adults, unfortunately, most of them were not having surveillance because they were lulled into this sense of they have mild disease because their neutropenia is mild. But their neutropenia might perhaps be mild because of these P53 mutations causing this compensatory rescue. We just don't know. We're still answering that question. It's really hard to know. Yeah. And most of them are not receiving G-CSF, not in a prolonged way anyway. It's not like the SCN patients, where they often need much higher doses. Schwachman patients respond to teeny tiny doses of G, and it's usually transiently needed.
Thank you. I have another question from Esther Bank. Does clonal hematopoiesis influence the risk for solid tumors in Shwachman-Diamond syndrome? And if I may add a second part of that question, do you think this might also be associated with some extra hematopoietic manifestations such as cardiovascular risk increase? Although that is very difficult to answer, but I would like to hear your opinion on that. Thank you.
These are fantastic questions. So the short answer is I don't know. But if I were to sort of explore these ideas further, I think that, let me see, the first one, I'm remembering the extra hematopoietic one. What was the first one? The first one was?
Solid tumors.
Mm-hmm. Solid tumors. So there have been reports of solid tumors, isolated solid tumors in patients with Shwachman-Diamond syndrome. But in order to prove predisposition, we have to show that the incidence is higher than what one might expect from chance. And there are so few of them, and you have to control for so many confounding factors that we've not been able to answer that. But it's not like in Fanconi or dyskeratosis congenita, where it's very easy to get those kinds of histories, and we personally see patients developing these solid tumors. They've been quite rare, even in the registry. They only have a couple. And in the published literature, also just a couple. So it's hard to know, though it is, of course, a huge concern. Anything that can predispose to P53 mutations would be very nerve-wracking. In terms of extra hematologic manifestations, certainly this ribosomal abnormality, both in the mouse model, be it through targeted CRE mouse models or more global, certainly the deficiency of Shwachman-Diamond syndrome affects multiple organ systems directly. So I think that's very clear from the models. The models, I will just say though, the models have been difficult to interpret because most of the models model loss, complete abrogation, knockout of SBDS. And in people, what they actually have is a hypomorphic mutation, most of them. It's a splice mutation that does allow a little bit of transcript, wild-type transcript and protein to be made. So they're basically hypomorphs, and it's been difficult to model that appropriately. Oh, I thought of one other thing to add to Jingwei's really great question about ribosomal stress and P53. One of the other differences in DBA versus Shwachman-Diamond syndrome is the level at which the hematopoiesis is affected. So in Shwachman-Diamond syndrome, we're affecting the stem cell, right? The stem cell, because you have multilineage abnormalities, lymphoid, myeloid, and the marrow becomes very empty. And as I showed you, the marrow empties out very early in life. It's amazing that these people can continue making these blood counts, in my opinion, with these cellularities that are like less than 30%, they're down in the 20% range for years. But DBA is normocellular, typically the bone marrow. And the defect is later in erythropoiesis. So it could be that it's the level of the stress within the hematopoietic compartment, as well as the hematopoietic milieu, because it's possible that the driver for somatic selection is very high when your marrow is empty and very stressed, as opposed to if your marrow is full of otherwise healthy hematopoietic cells, you just can't make red blood cells. It's possible that that milieu may not be as selectively favorable for these somatic clones.
I think it's an excellent analogy, and there were several cohort studies looking at somatic mutations in the Diamond-Blackfan anemia, which is the prime example of a ribosomopathy disorder, which actually did not find recurrent somatic alterations in these patients, especially in the patients who might have milder phenotypes or be clinically asymptomatic. So I wonder if there might be some other changes that lead to improved ribosome fitness at epigenetic or transcriptional level that we might be not detecting yet using the standard methods that we are applying.
It's such a great point. And this whole field of somatic rescue is sort of really burgeoning. I mean, your work and Jeff's work in SAMD9, SAMD9L has been really informative, right, in terms of somatic rescue. And then there's this POT1 mutation that can be acquired in the telomere biology diseases. You've got somatic rescue in your RPA1s. I think that we're seeing more and more of this and trying to distinguish which are compensatory and positive rescuing as opposed to those that are deleterious and drive malignancy is going to be very important.
Thank you. I also have one additional question on kind of the practical management of the surveillance in our patients. So let's say we have a 10-year-old child with Shortman-Diamond syndrome who comes for annual surveillance marrow. Based on your data, how would you change or what would you recommend? What type of assays would you recommend? Obviously, the assays that we have to order are have to be CLIA or CAP certified. So we cannot yet perform single cell DNA sequencing as an assay that will change our clinical management. But having said that, besides the cytogenetics, FISH, and the NGS panel, would you also recommend performing SNP array to look for the 17p UPD, or is this too early?
So 17p is something that we've seen certainly in these patients. We have not done that routinely. It's not a bad idea, though, to do that. Typically, we do all the other things that you just mentioned. We do something, I'm sure that you do this because you're a pediatric center, but the adult centers tend not to do FISH if the cytogenetics looked fine. And we found, we had to take it out of the paper because it was too long, but in our last Blood Advances paper, we found discordance between cytogenetics and FISH in some patients. And we think that it's partly because the marrow is failing. So which cells are giving you metaphases, right? We have no idea. They could be lymphocytes for all we know. So you could miss the monosomy 7 if it hasn't acquired sort of a proliferative advantage yet. And we found some discrepancy. So we always run FISH in addition to cytogenetics, regardless of whether the cytogenetics looked fine or not. And I know that a lot of patients are having trouble getting FISH analysis because I hear about this through the registry because of the recommendations through the adult pathology community that you don't need it if your cytogenetics are normal. But marrow failure is different.
Thank you. We have, Akiko, we have one additional closing comment by Dr. Jun Yang saying that it's actually interesting that we almost never see somatic rescue events in the ALL genomes, but most of the ALL germline driver, the germline causative mutations of ALL are in transcription factors, and they maybe do not cause profound stress, such as Shortman-Diamond syndrome.
Yeah, it's a great point. I think that we're talking about a failing marrow, right? So the things that we just discussed, Shortman-Diamond syndrome, DBA, SAMD9, SAMD9L, GATA2, they all cause marrow failure, which causes its own sort of stressors, but also a milieu that is conducive to outgrowth of these cells, as opposed to ALL, where perhaps it's different. Here, it seems like perhaps you need a proliferative driver. I would defer to you there, and I wish we could have an open discussion. I would love to hear your thoughts.
Okay, thank you very much, Dr. Shimamura, for this spectacular talk on the molecular mechanisms of somatic genetic rescue in Shortman-Diamond syndrome and related MDS. I would really like to thank you for your stamina. It's a second talk that you gave within 4 hours, second 1-hour lecture that is really excellent. And I would like to close this lecture and the Q&A, and thank you everyone for attending.
Thanks so much for having me.